Hey, it's Emma, and today I'm going to be talking a little bit about cell division, the cell cycle control system, and what happens when the cell cycle control system is thrown off. So in order to understand the cell cycle control system, you first got to have a basic understanding of how cell division works. So most of your cells, your body cells, divide through a process called mitosis. The cell spends most of its time in what's called interphase. Um, and interphase is basically when the cell is just growing and preparing for division. So there's three different phases of interphase. There's G1, which is when the cell is growing and functioning. The S phase, which is when the chromosomes are replicated in preparation for division. And then G2, which is when the cell is completing its preparations for division. The next phase is prophase. Prophase occurs when the duplicated chromosomes condense into pairs of identical sister chromatids, and that's what these are here. As you can see, these are identical chromosomes that are condensed. And then metaphase occurs when these chromatids line up at the center of the cell, of the centrosomes on opposite ends of the cell. Anaphase is when these when these chromosomes are dividing and going to opposite ends of the cell. Telophase is when, as you can see, this nuclear envelope is forming here. And then cytokinesis is when this part, the cytoplasm, actually separates, and we got two different cells that are now back in interphase. So the cell cycle is obviously a vital part of the cell's life and an organism's function, but how and why does it occur? And the system that regulates this is the cell cycle control system. And the cell cycle control system is defined as the molecules that systematically regulate the cell cycle's function in response to both internal and external factors. So what does that mean? At various points throughout the cell cycle, as you can see, this is a diagram of the cell cycle. Most of the time it's an interphase, and then there's a small portion of mitosis and a small portion of cytokinesis. But throughout this cycle, there's various checkpoints, and these checkpoints, the molecules have to receive a stimulus in order to allow the cell cycle to continue. And as you can see, the main checkpoints are G1, G2, and M. So the G1 checkpoint is actually believed to be the most important one because most of the time, once cells pass this one, they go through the rest of the cell cycle without a problem. But so this one must be passed before DNA synthesis can occur. The G2 must be passed before mitosis can occur. And then the M checkpoint has to be passed before cytokinesis can happen and the cell can actually separate. So what regulates this? Like, what happens at these checkpoints? Well, there's molecules that have to receive a stimulus to allow the cell to continue. And there's two types of main molecules here. One of them is protein kinase. And a protein kinase is an enzyme that can phosphorylate other proteins in order to activate or inactivate them. And this can stop or allow the cell cycle to continue. As you can see here, this is protein kinase. Uses ATP, releases ADP, and here it phosphorylates a protein, and that this phosphorylation gives the go-ahead to the cell to continue with the cell cycle. Okay. And this is where it sort of fits in here. This happens at the G1 checkpoint and the G2 checkpoint. I have it labeled here. Only the G1 checkpoint would have been redundant to put it here. But this is where that happens. So when this gives the go-ahead here at this checkpoint, then the rest of the cell cycle can continue, at least until it gets to the G2 checkpoint. And at the G2 checkpoint, there's actually a protein kinase that's called CDC2, and it's actually conserved in most eukaryotic organisms, from like humans to starfish. And so they believe that this is a conserved characteristic that can demonstrate evolution. So then the other type of molecule is called a cyclin, and cyclins are proteins that some kinases have to be attached to in order to be activated, in order to function. And these kinases are called cyclin-dependent kinases, or CDKs. And this is sort of a diagram that I made, just demonstrating like how a cyclin and a CDK fit together. This is the cyclin, this is the CDK, and the cyclin-dependent kinase can't operate until the cyclin is here with it. And this is where they fit in at the M checkpoint. There's a cyclin and a CDK that must allow the cell to go forward with cytokinesis. So, and the cyclin concentration in a cell can rise and fall depending on what part of the cell cycle it's at, and that's why it's called a cyclin, because the concentration cycles. So, as I mentioned earlier, these proteins can be impacted by internal and external stimuli. Internal signals occur, they're what they sound like, they occur inside of a cell to signal the protein to allow the cell to go ahead with the cell cycle or not. An example of this would be the transition from metaphase to anaphase in mitosis. So as you can see here, metaphase is when the sister chromatids line up down the middle of the cell, and they have to attach to the spindles here. Once they attach to the spindles, that gives the go-ahead for the cell to go ahead into anaphase, and sister chromatids begin to separate and move towards opposite ends of the cell. This does not happen until all of these sister chromatids are attached to the spindles. So if they don't attach to the spindles, the cell's not going to go into anaphase. And then as soon as they do, that sends the signal to go into anaphase. So that would be an example of an internal signal. And then external signals occur outside of the cell, basically what they sound like. And they're picked up by the proteins to send a signal to the cell to go ahead. An example of this is seen in blood platelets and fibroblasts. This here is a red blood cell. This is what's called a platelet, and a platelet is basically just a fragmented blood cell. But the platelets, when an area of your body is injured, the platelets release what's called a platelet-derived growth factor, or a PDGF, and this signals fibroblasts to go ahead for division. When the fibroblasts divide, a fibroblast is a connective tissue cell. And when the fibroblasts divide, this helps to repair the area that's been injured because there's more cartilage there. So what happens when cell cycle regulation doesn't occur? So when cell cycle regulation is impaired, that's how cancer forms. Cancer cells are cells that are unable to control their division. So they're basically dividing and dividing and dividing, and nothing is stopping them from doing that. Normal cells require growth factors in order to keep dividing. So this could be they have to have enough room to divide. There has to be room for them, or they have to have an anchor to attach onto. And when they don't have that, then they stop dividing. But cancer cells, for whatever reason, they don't respond to that. And scientists think that that's possibly because they are detecting growth factors that aren't actually there, or they can make their own growth factors, or that their entire control system is just messed up. They're still researching that. But so cells that uncontrollably divide are called cancer cells, and they form tumors. There's two types of tumors, and those are benign and malignant. Benign tumors are not able to spread to other parts of the body, so those can basically just be removed, and then the person is fine. But malignant tumors have the ability to metastasize, which means they can spread to different areas of the body, which is bad because then they can impair the functions of the part of the body that they've spread to. This is just a picture of how one cancer cell divides and divides and divides, and eventually is just out of control, and it forms a large mass called a tumor. Methods of treating cancer include radiation therapy, which, as you can see here, the UV rays are attacking or radiating into the tumor, and the hope here is that the DNA of the cancerous cells will be damaged so that it's no longer able to replicate the DNA and divide. So that's the idea behind radiation therapy. Chemotherapy is also another common method of trying to treat cancer. Chemotherapy is when you basically release a poison into the body that tries to kill cells that are actively dividing. And chemotherapy can be effective, but a problem that you run into with it is it attacks basically any actively dividing cell, so that includes healthy cells too, and that's why you see cancer patients undergoing chemotherapy that get sick and lose their hair and all of the negative side effects of it. So researchers are still looking into exactly what causes the cell cycle regulation systems to be messed up. They found that in many cases external stimuli can cause it, can throw it out of whack, and a good example of this would be how smoking causes lung cancer. And as you can see here, this is a pair of healthy lungs, and this is a pair of smokers' unhealthy lungs. And what's happened here is the tobacco smoke has and all the other chemicals in cigarettes has damaged the cells lining the lungs here, and when it causes cancer, it's because it messes up their cell cycle control systems. So in a lot of cases, external stimuli like smoking and other things can cause cancer, but it's not the only cause, and so that's why researchers are looking into it, because a lot of times it just seems to happen, although we don't think it actually just randomly happens. There is a cause, and that's what we're trying to find out, because that'll help us treat it and prevent it. So that's a little bit about cell division, the cell division control system, and I hope that's helpful.